So with time we have learned that plants have different way to defend themselves against pathogens. So most commonly used resistance was single gene or gene which had very large effect, and if you have one gene you get very clean looking plants, some spotting on the leaf, but pathogen would not multiply much. So that strategy from breeding side is very attractive, because you get many resistant progeny if you have such gene. But from pathogen side also, a small change is a little mutation in DNA of, for example, rust pathogen. What that would do, the protein which was being recognized, the pathogen protein which was recognized by the resistance gene produced protein, is no more recognized. So they don't recognize each other and plants become susceptible. So pathogen also learn how to evade by making a small change in the protein structure.
That's the gene for gene.
That's the gene for gene theory. And what we learned that as you commercialize varieties which have the single gene based resistance, in 3-4 in the Yaqui Valley, on the average 3 years was the life of a new variety, and resistance will break. So 3rd year is the year when variety is reaching in this area to its maximum coverage, and your resistance gene is just gone, broken. I mean, new race shows up, and that was like a constant battle. Here you got a good variety, and then you feel happy about that, and then you feel depressed, which is called boom and bust. So it's not only in the field, also breeders go through boom phase, and they should be ready for the bust phase. And how you are going to come back and come out of it, that means you need to have varieties which have other genes, a different gene for resistance. So this thing has been going on for a long time. And then we learned some early work done that there is another kind of resistance, adult plant resistance, which could involve genes which are probably defend in a different way. So they basically defend by slowing down the pathogens growth in the plant, and they do to all races same way. So no matter which race of let's say stem rust or leaf rust you apply, you will find that if plant has that gene, like the SR2, famous SR2 gene, you will see disease, but you will see much less disease. And usually if you have too much disease pressure, you may not see sufficient resistance in the plant if you have those genes which tend to have smaller effects than the major race-specific seedling kind of gene, what people often call. All these terminologies they use, which makes it more confusing to many people what they are talking about. So this concept of slow rusting, what we started to learn, that these genes also have ability to work together. So if I have one adult plant resistance gene, instead of seeing, let's say, 100% leaf area damaged or covered by rust, if that gene is present, I may have 80% coverage with disease, 60%, and in occasional case even 40%, but not more than that. But if you put two of these genes together, they get stronger, they get strength from each other, and then you might see that the plants which have two genes, now they have only 20% leaf area covered with rust. You put one more, it drops to 10 or 5%. You put a fourth one or a fifth one, you practically don't see any disease. So they give plant a different types of defense mechanism, which not only prohibits pathogen to be able to have successful infection, but at the same time, if rust has infected plant, it just affects the growth part of the pathogen. So pathogen starts to grow slowly rather than at normal pace. So you say the first infections are appearing much later than 10 or 14 days after the infection has occurred. It may be delayed by 4 days, 5 days, even sometime we see here 2 weeks. And that's a lot of delay in building up the disease epidemic, because for disease to build up, spores of the pathogen have to be produced in the field, in that field. So you start disease with few infections. With time, each infections are producing more and more and more inoculum, which are spreading by wind to the neighboring plants, other fields. But if you reduce the infections or the time when those spores will be released, or the new infections are releasing fewer spores, the progress, the whole dynamic of the disease progress has gone down significantly, and you escape out the epidemic. You may see some disease here and there with insignificant losses. And that is a change in the thinking, because these genes, which have different way of defending or giving defense to plant, it's like different kind of protection. You put your sunscreen, then you put a cap like me. It's a double protection against ultraviolet rays. Same way, you are getting different mechanism in plant to protect itself. So how do you select? See, when we learn that these genes, which in scientifically we call have additive effects, if you are together, the effect is much higher than when they are together. It didn't take much time for us to come up and say, okay, we will put a lot of disease in the field, and the plants which have much lower disease severity, much less infection. But the infection, when we look at the infection, let me pick this plant for example. It has fewer infection, but when you look closely, it looks like the type of it's a susceptible type of infection, but there it's much less. Let me try to look one more plant which has even hopefully I can find one to show you, but most here over here. Now you look them together. So in this one you have even fewer infections than this leaf, and they are even smaller, but they will produce some spores, but plants are also growing and growing towards maturity. So when we look at it visually, we see less infection. We say the morphology of infection means they are not causing these hypersensitive dead tissues, which is linked with the race-specific gene, where plants have to kill the cell to defend themselves. In this cell is more active mechanism. Plant is still alive, is trying to keep defending the invasion and growth of the pathogen. So it's slightly different. So rust and plants are every day battling, you know. The other mechanism is, rust needs green plant tissues. So if plants kill the area around where the infection has occurred, rust cannot grow any further. So, and that is called hypersensitivity. So the tissues around the infections are dead. So it's really very easy to make selection for this resistance gene if you are not confounded with the presence of race specific gene. If you have this major seedling race specific gene, then you don't see the presence of these genes. So we have come up with a nice strategy and made a lot of progress, that we don't mix the two types of resistance in the parents. So we pick parents which do not have the seedling gene or race specific gene, intercross them, and in the field as we select, we are selecting plants which have much lower disease pressure. So this principle was very successfully applied for leaf rust here. And in this, you probably heard during this conference, we were talking of a stem rust, we talked about yellow rust, but practically nobody talked about leaf rust, which is among the three rusts has much wider environmental range, because it likes from little cooler to little warmer temperature ranges. But people were not talking. Why? Because the CIMMYD and CIMMYD derived wheat varieties, majority of them have this kind of resistance. So we wanted to use this also for stem rust, and we are using for yellow rust a similar principle, that you don't want to have one gene with large effect protecting plant, because those genes do become ineffective. So we want several genes work together like a team and protect the plant. So very simple strategy, you know. So I believe in, we know problems are complex, but solutions need to be simple. If you want to implement, even in breeding, even in for farmers, you know, if you go to a farmer and say, I have these 10 things, if you will do, you will enhance your productivity. You think resource poor farmer will even understand those 10 things? You should go with 1 or maximum 2, this and this, and then they can implement it. Same thing we made it look like very simple, because now anyone can select, and that's what we are hoping. And because such resistance has shown that it can last 30 years, 40 years, 50 years even. SR2 has lasted more than 100 years. So we want more such genes to be identified in wheat, the modern wheats. They may be dispersed in different parents. You want to bring them together. And also in more primitive wheats, if we can find, we can bring them together and focus our breeding to that kind of resistance. So farmers don't go to the bust, and we don't go through the depression phase. Yeah, so one of the approaches to bring together these small effect adult plant resistance genes together and get the benefit from this team of genes. And same principle also applies if you use the race specific or the major genes. So each of these major genes or seedling genes or race specific genes, they are synonymous terms, tell you same thing. You just need one change in pathogen, so one gene is gone. But if in the same variety you have one more gene of that type, that gene will still give resistance to that race. So what happens is, if you combine 2 or 3 this kind of genes together, and they have to be effective to your pathogen population, you know, so they all should be giving resistance to the race population you have in your area. You can also enhance the longevity of these genes from 3 to 5 years to many years, because one mutation then is not enough to knock down 3 different defense. Let's say if I put 3 genes, I have 3 defense. So you go through 3 security check. One you were able to beat, but you don't beat the second one. So that spore which had the ability to beat one defense, that's where it stops. But then you also have to ensure that nobody else is releasing varieties which have those genes present as single genes. Because what happens if that happens, that this mutant spore, if it hits that variety, starts to multiply on that, then mutates again, and then it finds starts to combine or accumulate all this ability one at a time, and this we call a stepwise mutation. So this approach is also a good approach, using molecular markers. If you can pyramid 3 or 4 genes together, you can still get very effective resistance. The difference in the two strategy, to bring these major genes together, you do need molecular markers to select and ensure that they are together. Otherwise, whether one gene is present in field or 2 genes are there or 3, you don't see them. So you need this extra tool, and you need to have facility. You must have lab, and CIMMYT works with many small breeding programs. So, and they use CIMMYT germplasm. So what happens if I am sending wheat potential varieties which have 2 or 3 genes together? The day they make the cross with a susceptible variety, this combination falls apart. You have to rebuild, you have to reselect. And many of them don't have yet all the biotechnology facility, people to rebuild it. In contrast, there is adult plant resistance genes, because as you put them together, you see more and more resistance. So you can select them right there in the field. You do not need your sophisticated biotech facility. So this one becomes much more practical for many national programs to use than trying to bring the genes together.